Thanks, Ashley. Hello, everybody. It's nice to be here with you today. It's a nice afternoon here in Lincoln, and I'm really glad to be here and talk with you about a unique parameter called FM prime, which is a fluorescence parameter that we're going to talk about for about a half hour here. And I really enjoy talking about data, so I'm looking forward to talking about this with you. And I want to say at the outset that I've been using fluorescence methods for about the last 10 years to measure FM prime. And I was quite surprised when I came here to LI-COR and learned about how FM prime is prone to underestimation. And it sort of made me think about how I needed to go back and sort of reassess some of the data that I took over the last 10 years. And I was quite surprised and very interested by that. And what we'd like to do today is pique your interest in that same sort of technology. I'd like to say at the outset that we've recently published a paper entitled Closing in on Maximum Yield of Chlorophyll Fluorescence Using a Single Multiphase Flash of Subsaturating Intensity. And within that paper, you can get a lot more of the gory details about what we're going to be talking about today. And as Ashley mentioned, you guys can send in questions to our panelists and comments as well, and we would really like to encourage you to do that. We would sort of like this webinar to be sort of a back and forth between you and I, and when I say I and you, I mean people all over the world. So this is a really unique forum to communicate with one another, and let's all take advantage of that. So what I'm going to do today is I want to briefly review photosynthesis so that I can talk about FM prime in that context. And then I'm going to talk about two different ways of measuring FM prime, one of which is referred to as a traditional method, and the other is the multiphase flash method that we're going to talk about. And then I'm going to end with some test cases whereby we favor the multiphase flash method over the traditional way of measuring FM prime. So just by way of introduction, you all know that photosynthesis is responsible for removing carbon dioxide from the atmosphere, producing the sugar molecules by which plants take physical form. And in the process, the oxygen that you and I are breathing is produced. Today we're going to be talking about those processes at the level of the leaf, for it's deep within leaves that the specific sites of oxygen and gas exchange with the atmosphere. For example, here is a cross-section of a leaf. The upper and lowermost layer of cells are referred to the epidermal layers. In between the epidermal layers are two other cell types, one of which is called the palisade parenchyma, underneath which is a less organized layer of cells referred to as the spongy mesophyll. In all three cell types reside the specific sites of gas exchange with the atmosphere, the chloroplasts. Chloroplasts are double membrane organelles, enclosing an aqueous compartment referred to as the stroma. And it's within the aqueous compartment that the enzymes reside that assimilate carbon dioxide into the sugar molecules. Now the first question that I'm going to ask is a gimme. Can somebody go ahead and write in, where does oxygen evolution occur in the chloroplast? Specifically, where does it occur? So again, this is the double membrane. Inside the aqueous compartment we have this complex network of membranes referred to as the thylakoids. And so the question is, where exactly does oxygen evolution occur in the chloroplasts?
We'll give everyone just a moment to type in their answers. And if you're wondering where you can submit your answers, there is a box, a questions box in your control panel. So just go ahead and expand that and type it in right there, and we will see everyone's answers pop up for us here.
And for you leaf experts, you know out there that there are other different types of cell types in different leaves. So this is just a model just to introduce the concept of where photosynthesis happens.
Tom, it doesn't look like we have any guesses, so maybe you should just go ahead and give everyone the answer.
Okay, sure. Thanks, Ashley. Yeah, so oxygen evolution, the specific site of oxygen evolution is the thylakoid membranes. And in a nutshell, photosynthesis can be described by the evolution of oxygen and the consumption of CO2. And the reactions that mediate these processes are coupled, or they're linked. For example, shown here is a cross-section of a thylakoid membrane. Embedded in the thylakoid membrane are all of the proteins and cofactors that mediate the conversion of light energy into chemical energy. And so, for example, absorption of light at photosystem 2 results in the oxidation of water, the evolution of the oxygen that you and I are breathing, electrons as well as protons. The electrons are transferred through a series of intermediates, one of which is the plastoquinol. the cytochrome B6F complex, and plastocyanin. Upon absorption of light at photosystem 1, the electrons are subsequently used to reduce NADP+ to NADPH. Now, as I mentioned, oxidation of water results in protons being deposited inside the lumen. However, the oxidation of the quinol species at the cytochrome B6F complex also results in protons being deposited into the lumen. This generates an electrical chemical gradient of protons, or proton motive force, and those protons, essentially, they want to move down their gradient, and they do so through the ATP synthase. And in this way, their movement is coupled to the formation of ATP. And in this way, light energy is converted into usable chemical forms of NADPH and ATP. Depicted here is a very simple schematic of CO2 assimilation, almost too simplistic actually, but I just simply want to point out that during CO2 assimilation, carbon dioxide is combined with ribulose 1,5-bisphosphate, ultimately generating a pool of triose phosphates, glyceraldehyde 3-phosphate, from which plants ultimately take physical form. I just want to emphasize that the point of this slide is that the enzymes that mediate this carbon dioxide assimilation require energy, and the energy in this case is in the form of ATP and NADPH. Now, all of these reactions are coupled or linked in the sense that the light reactions produce the chemical energy that the enzymes use to assimilate CO2 into sugar phosphates. And in essence, photosynthesis can be broken down into energy converting reactions and energy consumptive reactions. And today we're going to be predominantly dealing with the energy converting reactions. For several decades, pulsed amplitude modulation chlorophyll A fluorescence has been used to study many different aspects of light energy conversion. And so I want to briefly review that technique here. So depicted is a low-intensity modulated or pulsed light source. Illumination of the thylakoid membrane with that pulsed light source results in fluorescence being elicited predominantly from photosystem 2. The resultant amplitude of the modulated fluorescence is referred to as the minimum fluorescence yield, and I'm going to denote that as F here. Now, simultaneous application of an intense light flash results in some unique things within photosystem 2, and I want to ask a question about that. That will be our first poll question. Ashley, would you mind putting that poll question up?
Sure, it should be up on our screen now.
So the question is, will the modulation light elicit more or less fluorescence when the saturation flash is simultaneously applied? And so while you're thinking about this, what I want to point out is that during application of the high-intensity flash, first of all, the intensity of that flash could be upwards of 10,000 micromoles per meter squared per second. However, the intensity of the modulated light doesn't change. So take that into consideration when you're thinking about whether or not the fluorescence yield is going to increase, decrease, or stay the same when the high-intensity flash is given. How are we doing, Ashley?
So we're at about 24% voted. So those of you who aren't quite sure how to vote, go ahead and just click the button right next to the word that you'd like to use as your answer, and it should submit for you.
Don't be shy. Go ahead and push that button.
We're right at about 53% at the moment. We'll wait just a little bit longer to give everybody a chance to get an answer in here.
You guys are doing great. So some of you who use this technique, you may not have thought about it in terms of the different intensities of the light sources that are used to mediate this technique. So.
All right, we're at about 76% of the audience having voted. So I'm going to go ahead and close the poll and show everyone our results.
Great.
So we have about 77% of our audience answering more and about 23% answering I'm not sure. So we'll go ahead and let Tom explain the answer to you guys.
That's a pretty good outcome. Yeah, so what the high intensity light flash basically modulates the redox state of the reaction center of photosystem 2 in such a way that even though the modulated intensity is not changing, the fluorescence yield that's elicited is significantly more by the modulated light. The amplitude of the resultant fluorescence yield is referred to as the maximum fluorescence yield, and it's denoted as FM prime when it's obtained in a sun-exposed leaf. So good job, everybody. Looked like most of the people got the right answer, so great. Now, because the rise to FM prime is specific to photosystem 2, And because there are other processes within PS2 that can compete with fluorescence for dissipating absorbed light energy, for example, electron transfer, mathematical models have been developed that allow estimation of FM prime to provide information about all kinds of other processes that happen that are associated with photosystem 2. In essence, FM prime is a core parameter for estimating a variety of other photosynthetic phenomena. For example, FM prime is used to measure the quantum yield of PS2 electron transfer, or phi PS2. Phi PS2 itself serves as input in an expression for quantifying the rate of electron transfer through photosystem 2, that's shown here. Comparison of electron transfer with gross CO2 assimilation can provide information about the electron requirement of CO2 assimilation. Estimates of electron transfer can also be used in conjunction with gas exchange parameters and the LI-COR model of photosynthesis in order to obtain estimates of GM, which is mesophyll conductance. And mesophyll conductance is a measure of the pathway by which CO2 moves from the substomatal cavity to the site of carboxylation in the chloroplast. Estimates of GM can then be used to back calculate the concentration of CO2 in the chloroplast, or CC. Photosynthesis response curves based on CC can provide accurate estimates of rubisco-kinetic parameters, which subsequently are used in climate and crop modeling systems. Estimates of FM prime are also used to measure energy-dependent quenching, or QE. Comparison of QE with electron transfer can provide information about how light capture is regulated in response to fluctuating environmental conditions. And lastly, FM prime is used to measure non-photochemical quenching, or NPQ, which is an important stress parameter. So while FM prime can be used to measure all of these different photosynthetic phenomena, a problem arises because FM prime is prone to underestimation. And this error can be propagated to estimation of all of these other parameters. And so what I'd like to do now is show you how underestimation of FM prime can be inferred. So shown here is a series of increasingly intense, what I'm going to call traditional rectangular flashes. And these rectangular flashes are shown increasing in intensity between 3,400 to 8,500 micromoles per meter square per second. Now remember that depending upon where you are on the Earth and the time of year, full sunlight is somewhere between 2,000 and maybe 2,300 micromoles per meter square per second. And so these intensities are purely artificial and are never experienced by plants in nature. The maximum intensities of these rectangular flashes are held constant for between 0.5 to 1 second. And so while they are quite intense, it's only for a short duration of time. Now I'm going to be referring to the fluorescence yield obtained during the maximum portion of the rectangular flashes as the apparent maximum fluorescence yield, or AFM prime. And so this leads into our second poll question. Ashley, can you take care of that?
Yes, give me 1 second and
Sure.
I will get it popped up. All right, everyone should be able to see the question now.
So the question is, will apparent FM prime increase linearly, sigmoidally, or hyperbolically as a function of increasing flash irradiance? So that's essentially asking what's going to happen to the values of apparent FM prime as these flash intensities increase. Will it exhibit a linear, sigmoidal, or hyperbolic curvature?
All right, we're at about 6% voted right now, so we'll give everyone some time to think this through.
Just for those of you, if you would have asked me this about 4 years ago, I would have sat here for a half hour and not clicked one of those buttons. So I'm not surprised that we have a low percentage. It's sort of a trick question.
It's jumping up.
Oh.
So we'll give everyone, we're at about 47%.
Oh, wow, great.
We'll give everyone just a little bit longer to submit an answer.
Great.
And go ahead and guess. If you don't know, it's okay.
40% is pretty good.
All right, we're close to 60% now, so we'll take these answers and show everyone the results. So it looks like about 10% said linearly, 20% sigmoidally, 40% hyperbolically, and then 30% we're not quite sure.
Wow, that's great. That's a great response. All right, well, let's look at the data. So shown here are experimental estimates of apparent FM prime plotted as a function of this increasing series of increasingly intense rectangular flashes. And for those of you who answered hyperbolically, sure enough, there you see it. So what's being shown here is that the apparent values of FM prime increase in a hyperbolic manner in response to the increasing flash intensity. Now, because these values are increasing in a hyperbolic manner, that implies that none of these values really represent the true value of the maximum fluorescence yield. And so, for example, historically, traditionally, true estimates of FM prime have been assumed if the apparent values approached saturation, as shown here by this hypothetical data set. Now, you might be saying, well, one way to increase the value of apparent FM prime would be to simply increase the flash intensity, and that is one way. However, there are problems with that from a instrumental point of view as well as a physiological point of view. And fortunately, we've got a couple more answers?
Actually, we have a question.
Oh, great.
Rodrigo is asking, what will happen with PS2 if the flash was longer than 1 second?
That's a good question. So what I mentioned is that there are problems with the flash intensity from an instrumental point of view as well as a physiologic point of view. And let's deal with the instrumental point of view first. So if you're applying voltage to an LED to cause it to illuminate, that LED is going to heat up, and the dyes in those LEDs are going to lose their efficiency at those high intensities. And so there are problems in one sense with keeping the LEDs at a constant intensity, and if you increase the duration of the saturation flash, you're going to run into those types of problems. Now that's instrumentally, that's just one example. Now from a physiological point of view, as I mentioned, these intensities are never seen by plants in nature, and they can actually cause harmful side reactions within the reaction center of photosystem 2, some of which might involve formation of harmful forms of oxygen. And so therefore it's best to try and keep these saturation flashes within a short time span. That's a great question, Rodrigo. Thanks for asking that. Okay, so fortunately for the scientific community, some very clever scientists out of Stanford developed a technique that takes advantage of these values that are approaching saturation, that are closest to saturation, in order to derive a method that allows estimation of fluorescence yield at infinite irradiance. So what I'm going to show here in a minute are these three values of apparent FM prime plotted as a function of the reciprocal of the flash intensity. And so take note of the fact that as the x-axis approaches 0, it does so at infinite irradiance. That's the first thing to point out. The second thing to point out is that while these values have a little bit of hyperbolic curvature when plotted in this manner, when they're plotted as a function of the reciprocal of the flash intensity, they can be well approximated by a linear function. And this serves as a basis for estimating fluorescence yield at infinite irradiance by linear regression and extrapolation to the y-intercept. The resultant value is referred to as the extrapolated FM prime, or EFM prime, and has been shown by the scientists that developed this method to be a closer approximation of the true value of FM prime. What I'm showing here are these same values of apparent FM prime as a function of flash intensity, but I've normalized the entire data set to this extrapolated value, which we are interpreting as a true value of FM prime. And what you can see is that depending upon the flash intensity, the apparent values underestimate the true value of FM prime by between 4 and 11%. So now, while this method is capable of providing an accurate estimate of true FM prime, it requires the application of several variably intense rectangular flashes, each of which must be separated by 1 to 2 minutes so as to avoid damage to the leaf, which limits throughput of the approach. Therefore, at LI-COR, we've developed a new method of more rapidly estimating FM prime at infinite irradiance. And that method is based on what's referred to as a multiphase flash fluorescence. And what I'm showing here are the changes in irradiance and fluorescence yield as a function of time during a typical multiphase flash, which consists of 3 contiguous phases of change in irradiance. So, for example, during phase 1, the irradiance is increased from a background intensity of 2,000 up to about 7,800 micromoles per meter squared per second for a duration of 300 milliseconds. And so phase 1 is very similar to a traditional rectangular flash. During phase 2, this maximum intensity is linearly attenuated by 60% for a duration of 500 milliseconds. During phase 3, the irradiance is returned to the phase 1 level for another 300 milliseconds, after which the pulse is terminated. So while the multiphase flash happens within a 1 second, with about a 1 second period, very similar to a traditional rectangular flash during which the irradiance is held constant, the changes in irradiance during a multiphase flash are clearly more dynamic. The changes in fluorescence yield are equally dynamic. So during phase 1, the fluorescence yield increases to an apparent maximum, or AFM prime. During phase 2, the fluorescence yield decreases hyperbolically, only to recover to the phase 1 level during phase 3. The phase 2 fluorescence yield can be used to derive fluorescence yield at infinite irradiance. For example, shown here is the phase 2 fluorescence yield, here, plotted as a function of the reciprocal of the phase 2 irradiance. Recall that the x-axis approaches 0 at infinite irradiance. Linear regression and extrapolation of the fluorescence yield over the highest 25% of the change in irradiance results in an extrapolated value that was about 10% higher than the maximum apparent value. And once again, we interpret this as a more or closer approximation of the true value of FM prime. And so while this is conceptually very similar to the traditional method of measuring fluorescence yield at infinite irradiance, it does so within the period of a 1 second flash, as opposed to the series of applications of rectangular flashes by the traditional methodology. The multiphase flash can be very informative. What I've depicted here is a series of increasingly intense multiphase flashes ranging in intensity from 5,000 to 13,000 micromoles per meter square per second. Ashley, this is the point at which I want to have the third poll question brought up. Could you do that?
All right, I will launch that right now. Everyone should be able to see the question.
So while you're thinking about this, just remember that, as I mentioned, the fluorescence yield that's measured during phase 1 is the apparent value. During phase 2, we have a hyperbolic change. And during phase 3 is another type of, another measure of apparent FM prime. So when you're considering how many values of apparent FM prime could be obtained with all 5 of these multiphase flashes, keep that in mind. Let's test your knowledge of the multiphase flash method.
Looks like we've had a few people go ahead and vote, so we'll give everyone just a couple more moments to choose an answer.
This is a little tricky, but I think you guys are pretty bright. You guys are pretty sharp. You've answered most of the questions right on the money.
All right, we're at about 1/3 of our audience, so let's see what we can do to get about half of you guys answering.
Okay, well, let me give you a hint then. We've got 5 multiphase flashes. You can get 5 different measurements of apparent FM prime during phase 1, 5 different values of apparent FM prime during phase 2. Now the question is, how many values of extrapolated FM prime can be derived from each one of these multiphase flashes?
All right, we're at about 56%, so
great.
Rising. Yeah. I think that might have helped some of our audience.
that should have helped.
Yes, I think they got it. All right, we'll go ahead and close our poll question and let you guys see the answers. So 17% answered 5 and 1, 33% 1 and 5, 42% 10 and 5, and a few of you, 8%, said you're not quite sure.
Ah, great. You guys are doing great. Yeah, so the answer is 10 and 5. So with all 5 of these multiphase flashes, 10 different measurements of apparent FM prime can be obtained, and 5 different measurements of extrapolated FM prime. So let's look at those values and see the type of information that you can get from that. So what I'm showing here are the values of apparent FM prime during phases 1 and during phase 3 of these multiphase flashes, plotted as a function of the increasing phase 1 intensity. Also shown are the extrapolated values of FM prime associated with each one of these multiphase flashes. Now the other thing I want you to be aware of is that I've normalized the entire data set to the extrapolated value obtained at 13,000 micromoles per meter squared per second. So the first thing to point out is that the extrapolated values remain pretty constant between about 7,000 and 13,000 micromoles per meter squared per second. And so what this implies is that an accurate measure of FM prime could be obtained at an intensity as low as 7,000 micromoles per meter squared per second when used in conjunction with the multiphase flash method. Second thing to point out is that depending upon the phase 1 intensity, the apparent values underestimate the true value of FM prime by between 8 and 12%. And lastly, the very slight differences between the apparent values during phases 1 and 3 imply that there was no significant induction of these harmful auxiliary reactions quenching fluorescence during the multiphase flashes. And so there's lots of different types of information that can be obtained during a multiphase flash. The multiphase flash method can also be used to derive accurate measurements of fluorescence. And so what I've shown here are a series of estimates, fluorescence estimates, FM prime, phi PS2, and MPQ, as estimated by the multiphase flash or the rectangular flash method. And so the line means if a value of 1 on this axis means that the corresponding values are exactly the same. And so therefore the way I've written the ratio, if the value falls above the line, that means that the multiphase flash derived value is greater than the value based on the traditional method. And what you can see is that in both maize and sunflower, as a function of increasing background light intensity, the multiphase flash derived estimates of FM prime were invariably higher than their apparent their rectangular flash counterparts. Now remember that FM prime is used to measure both MPQ and phi PS2. Therefore, what you can see is that in both maize and sunflower, the multiphase flash derived estimates of phi PS2 are invariably higher than their apparent than their rectangular flash counterparts. And in contrast, the multiphase flash derived estimates of MPQ were invariably lower than their rectangular flash counterparts. And so what these data imply is that the traditional way of measuring FM prime by rectangular flashes underestimates FM prime, and that singular act of underestimating FM prime results in an underestimation of phi PS2 and an overestimation of non-photochemical quenching, both problems of which are solved by using multiphase flash methodology. The multiphase flash method can also be used to accurately measure photosynthetic coupling. And recall that electron transfer is estimated as the product of phi PS2, light intensity, and some correction factors. Phi PS2 is estimated using FM prime, and the value of FM prime can either be the extrapolated value based on a multiphase flash or the apparent value based on a comparably intense rectangular flash. And therefore, the value of FM prime used as input in the equation for phi PS2 is ultimately going to affect the value of electron transfer. Now, we don't have a poll question for this, but let me test your guys' ability and ask you, what's going to happen to the value of electron transfer if FM prime is underestimated?
All right, we'll let everyone just type those answers in our questions box then. So we'll give everyone just a couple seconds.
So it should be pretty straightforward. So basically you're looking at, you have to hypothetically think, if the value of FM prime is lower than the true value, how is that going to affect phi PS2? And furthermore, how is that going to affect the estimate of electron transfer based on that? How are we doing, Ashley?
I think you may have stumped our audience.
Okay, well, let's take a look at the data then. So what I've shown here are estimates of electron transfer plotted as a function of gross CO2 assimilation in maize. The dark spheres correspond to the data set where electron transfer was based on MPF methodology. And what you can see is that a linear fit of these data resulted in a slope of about 4.7 electrons per CO2, and the intercept was not statistically different than 0. And this slope of 4.74 is essentially what's predicted from theory. In contrast, the open spheres correspond to the data in which electron transfer was based on rectangular flash methodology. And what you can see is that the slope of a linear fit of that data set was 2.94 electrons per CO2, which is essentially meaningless. For those of you who are familiar with making these sort of comparisons, the absolute, at least in maize, the absolute minimum number of electrons per CO2 is about 4.7. And so what these combined data imply is that underestimation of Fm prime by traditional methodology can result in an erroneous estimate of photosynthetic coupling. And more importantly, that this problem can be overcome by use of multiphase flash methodology. The multiphase flash method can also be used to accurately measure mesophyll conductance, which for those of you who don't know about mesophyll conductance, it's a very hot field right now, and it's being studied intensely by groups all over the world. And one way of measuring mesophyll conductance is based on combining gas exchange parameters, the LI-COR model of photosynthesis, and fluorescence-based estimates of electron transfer. And so shown here are estimates of mesophyll conductance in sunflower as a function of increasing background light intensity. And the upper panel represents estimates of mesophyll conductance that were based on rectangular flash-derived estimates of electron transfer. And I want you to pay attention to the scale between 0 and I think it was 4.5, and 0 and negative 4.5. And these data are highly erratic. They're very, very noisy, and they're well outside of the range of literature-reported values of mesophyll conductance. In contrast, the lower panel represents estimates of mesophyll conductance based on multiphase flash-derived estimates of electron transfer. And the first thing I want to point out is the scale. This is between 0 and I think 0.75, so the scale is very magnified. And what you can see is that the data are nice and constant, and reasonably noisy, and well within the range of literature-reported values. And so taken together, these combined data imply that underestimation of Fm prime by traditional methodology can result in erroneous estimates of mesophyll conductance. And once again, this is a problem that can be overcome by use of multiphase flash methodology. So in summary, what we wanted to do today is introduce, for those of you who are not aware of it, a unique parameter referred to as Fm prime, which is a fluorescence parameter. And furthermore, that accurate estimation of Fm prime is important for correctly understanding all kinds of different aspects of photosynthesis. So for those of you who are measuring just fluorescent stuff, it's important. But as we've also tried to show you, is that for those of you who are combining fluorescence and gas exchange, it's very important also in that field. And lastly, what we've tried to show is that the multiphase flash methodology leads to more accurate estimates of Fm prime as well as the other Fm prime related phenomena. And at this point, I'd like to acknowledge all the scientists behind this. Shannon Loriot was basically spearheaded the entire project and has overseen the project from start to finish. Drs. McDermott and Wells were the supervisors and the intellectual drivers for the project. And I'd like to acknowledge the engineers who very cleverly helped design and build some home-built fluorometers early on that helped us to test this technique. And lastly, I'd like to thank Dr. Hugh Earle and Dr. Pat Morgan for their helpful comments throughout development of the method. And I want to end with just reminding you that we're doing a second follow-up webinar next week, and some of the information in that webinar is going to be redundant. However, we're going to go into a little bit more of the detail of the underlying physics of the phenomena. And for example, we developed a model to simulate multiphase flash dynamics, and we feel like we have a very good understanding of the underlying changes in the reaction center redox dynamics during a multiphase flash, and we're going to talk about that next week. And lastly, I just want to end with if there are any questions that you were not able to ask during the webinar, you can send those questions to Rod Madsen, who is our product manager, or you can send them to myself. And with that, I believe we might have time to take a couple questions. Ashley, how are we doing?
Yeah, we're about 6 minutes past the top of the hour, so we're a little bit over for time, but we've got quite a few people still hanging around with us. So we'll give everyone just a couple of seconds to type in those questions if they have them. Right now, I don't have any for you.
Okay.
But I wanted to thank everyone for their participation today. It was really fun to see your answers as we move throughout the presentation. And that we will also be sending information on how to access a recording of this webinar really soon. So watch your email. Or you can go to our website, LI-COR.com/env/webinars for details on how to watch this replay. Our panelists are answering questions, so stick around if you have a question that hasn't been answered yet, or feel free to give us your contact information if you'd rather receive an email response. So we'll be taking questions for just a little while longer, and we will stick around until all of those have been answered.
Yeah, don't be shy. Go ahead and ask those questions.
It does look like we have a couple questions that have come in. Mark says, could you please clarify your comment regarding the minimum number of electrons needed per CO2 for maize?
Yeah, so that's referring to this. If you do the number crunching of the stoichiometry of electrons transferred through the electron transfer components, as well as the protons and the coupling of those protons to the formation of ATP. In maize, well, actually in plants in general, the minimum number of electrons turns out to be 4 electrons per CO2 assimilated. In maize, there's a little bit of an extra requirement that's associated with the energy needed to concentrate CO2, which is the one of the unique aspects of C4 plants. And so that number is a really important quantitative number because it's a benchmark. So if you do a light response curve and you see a slope, at least in maize, a slope different than about 4.6, 4.7, then depending on which way that's deflecting, either upwards or downwards, then that tells you that something could be wrong with your measurement, or it could tell you that your plants are under stress, because under stress there's going to be a shift in the slope. It's going to increase. And so that benchmark number of 4.7 is very important for comparison of stresses and techniques. And we have one other question. Go ahead.
Yeah, we have a couple more in the queue, and our panelists are answering a few of the questions as well.
Okay.
The next question I have is from Whitney, and it is there a situation where it is not appropriate to use multiphase flash and to use the rectangular? Or is it now the recommended way to determine FM prime?
That's a great question. And I'm going to go ahead and back up a couple of slides to answer that. So bear with me. Ashley, can they still see what's on the screen? Okay, great. So I'm going to back up a few slides here. Please be patient. Okay, this slide here. So what we recommend in the paper that I highlighted at the beginning is that when you're initially doing an experiment, that you generate a graph just like I've showed here. And you don't necessarily have to normalize it to the maximum extrapolated value, but what we recommend is that you do an experiment where you look at the hyperbolic curvature of the apparent values and the values of extrapolated FM prime. And I have to be honest, in the 4 years that I've been studying this technique and developing it, I've only seen the apparent values come together with the extrapolated values one time in one species. Invariably, what I see is something like this, where the apparent values, even at the maximum intensity setting, are somewhere between 4 and, depending on the species and depending upon whether you're in a greenhouse or field-grown plants, the maximum value is usually between 4 and 10% lower than the extrapolated value. And so what you're going to want to do when you're using this technique is you're going to want to generate a curve like this under low light intensity as well as a high background light intensity, and determine whether or not you even need to use the multiphase flash method. And but just to reiterate, only one time in 4 years have I seen these values come together. And if they come together, so Ashley, I'm going to go ahead and use the pencil. Is that okay?
Sure.
All right. I'm going to use the pen. So if the apparent values do something like this, I'm trying to draw them hyperbolic, but they come together up here at the extrapolated values, then what that would mean is that the apparent value obtained at this intensity setting of 13,000 is an accurate measurement of true FM prime. But like I said, I've only seen that once. Now the second thing about the multiphase flash method is that just because you can estimate the apparent value at this really high intensity, there are several things to consider. One is that these really intense flashes can be really damaging to the leaf. They can suck energy out of your instrument, and so even though you may see something like this where you see the values come together like this at the highest intensity, you can still back off the intensity when you combine it with the multi-phase flash method and use much lower intensities and you can get accurate estimates of FM prime while using lower intense flashes. So that conserves battery power. It also prevents damage to the leaf. And so yeah, so the long story short is we recommend that you measure this sort of graph. You create this sort of graph when you're deciding whether or not to use the multi-phase flash method. And we have one more question.
Yeah,
good question, by the way, Whitney.
Yes, I have one more question from Chris, and it is, how do we implement the MPF technique?
How do we implement? Yeah, so if you've bought a 6400 XT or above, the instrument automatically comes with the multi-phase flash method. Now, for those of you who don't have an instrument in front of you, you're just going to have to sort of remember how to manipulate your way through the menu screens. But in new measurements mode, if you go to menu level 2 and you go to, if you go to menu level 8, F2, that you'll be able to modify your fluorescence parameters, and all instruments that come out of LI-COR automatically default to using a rectangular flash. However, if you put your cursor next to where it says rectangular flash and you hit edit, it will automatically cause the instrument to go from rectangular flash to multi-phase flash. And furthermore, the instrument has already been set up with optimized ramp depths and ramp rates, and so you shouldn't have to tinker too much with tuning the multi-phase flash. We've set the instrument up so that it's optimized. But once again, make sure that when you use the technique that you generate this sort of graph. Doesn't take very long to do, and I would do it at a couple of different light intensities, for example, a low light and a high light intensity. And I will use this opportunity to say that we've observed that this is really important for field-grown plants. It turns out we haven't really, we're exploring it, but we haven't really figured out exactly why this is the case. But under field conditions, the difference between the maximum apparent value and the extrapolated value is most enhanced. And therefore, for those of you who are doing field measurements, you're really going to want to take a look at this if you're interested in measuring stuff that has fluorescence values in it.
Thank you, Tom. That was a lot of valuable information.